Good morning, everybody. All right, so today we will continue on our journey through translation. Just checking the. And so essentially, we're a bit ahead of time, to be honest. We've made pretty good progress, um, and we potentially have. I wonder if might actually be changing the schedule to have the exam next Friday because it's been schedule's been all kind of messed up for a bunch of reasons. Um, and that would tie in a lot better with the other class as well. So I'll yeah, so essentially the reason for doing that would be that let me just show you. Oh, no, love. So where are we at? Right, so the exam would have been around here apart from the craziness of Snowmageddon and various other issues. And so if we move, essentially we're already at translation here, so we're actually making real good time. And so if we put it here, that still leaves us potentially a couple of catch-up days, which would be nice because there's a lot of material here, and that would also give us a catch-up day for the last exam as well, which would I think would be a benefit all round, rather than having a whole week off, which I don't think is really necessary. So I'm going to change that and make yourself a note. Send that round after class. So that still gives you a whole week to prepare. It shouldn't really change your preparation too much. Okay, so where were we at? All right, we were kind of around here from memory. So essentially, and I'll send an announcement around when I upload a new schedule as well, so that everybody's on board. Okay, so anyway, we kind of left off with the basic properties of amino acids and polypeptides and proteins as well. And I can't remember if I went into this with you because I think we stopped on this this slide talking about the individual properties of amino acids. Well, obviously, those come together to form a protein, right? And all of those individual properties, essentially, the sum of that is the structure of the protein. With protein, structure equals function, right? So these very much kind of get at the function of the protein. And one thing I don't think I got round to last class, and correct me if I'm wrong, but one of the really interesting things about all of these is that typically in enzymes, even things like RNA polymerase and transposase and various catalytic enzymes that glycolysis and the like, typically their active sites have several of these basic and acidic amino acids present in the pocket, right? So in the actual kind of three-dimensional shape where the reaction takes place, quite commonly there are positively and negatively charged amino acids involved. And the reason for that, is that these are able to interact with the substrate, typically to hold it in a certain position, which allows the reaction to go ahead, which makes the reaction more favorable, essentially. So anyway. And also, so this is like a cartoon version of a protein. I've showed you several other pictures of proteins. Actually, did I show you? I'll show you in a second. I'm not sure if I show you this one. I did the other class. So essentially, this is what's called I think it's called a string and ribbon model of protein structure. And obviously it doesn't look like there's a bunch of arrows in there, right? Or the like, because that would be really weird. But essentially what this is showing are this is the 3D, the tertiary structure, right? Which is also three-dimensional overall, right? So what that protein actually looks like, its shape, is its tertiary structure. But that's made up of, and so here you can see this is the polypeptide kind of chain kind of going through here, and then it goes through all over the place. It's actually kind of a little bit like a game trying to find out where it goes, right? And so that single string of amino acids is called the primary structure, right? The polypeptide chain. The secondary structure are these things here. So these are alpha helices, or is this an alpha helix, right? Because it creates a spiral. And they actually do look like that in real life. They are very kind of spiral, twisted forms. It's my daughter sneezing. You also have, so these are able to move in kind of most directions, right? But they're not very compressible. These are anti-parallel beta pleated sheets. And they're called pleated sheets because they are kind of flat, kind of ruffled sheets. And so these have a lot of flexibility in the flat plane and very little flexibility in the side plane, right? And so you can see how these are going to determine the structure of the protein and how that protein moves or not, as the case may be. And then you have these kind of unstructured regions called loops. And really they allow these other secondary structures to assume the correct position within the protein. And also they're very important for the protein moving, right? So most proteins move, not all of them, but most of them change shape, right? Kind of move from typically from one shape to another, and not a huge movement necessarily. Myosin is the exception. That has a very large range of movement, but they do move and flex, and that's typically very important for their function as well. And so my current fave protein used to be hemoglobin, but actually I think ATP synthase has won that particular contest. So this is what ATP synthase looks like. So this is the protein or collection of proteins that generate ATP using a proton gradient. This is an inner membrane of mitochondria. And so this is a protein that's made up of kind of 4 different groups of proteins, right? There's the stator, which holds everything together. There's the channel, the axle, and the head. And this is actually what makes ATP synthase, ATP, sorry. And so remember I was talking about these beta pleated sheets, not the beta pleated sheets, these alpha helices. And you can actually see them here, right? So this is a barrel of alpha helices, right? And it's in a membrane, a phospholipid bilayer, right? And can anybody remember what's the property? This is kind of more Gen Bio 1 stuff. The property of the inside of a phospholipid bilayer or plasma membrane. Is it hydrophilic or is it hydrophobic?
Hydrophilic, right?
Opposite. Good guess though.
Hydrophobic, or the outside's hydrophobic?
The inside is hydrophobic. The outside is hydrophilic. If you think about our kind of cellular environment, right, it's largely water-based. You know, the cytoplasm is aqueous. Outside the cells is typically aqueous in some regard, like your blood, for example. But the inside, so the outside of the membranes have to be hydrophilic because they interact with that water. And the inside is hydrophobic, essentially because that's what allows these membranes to assemble. If you can remember, they're like basically stick figures with their legs kind of touching, and it's the legs which are the hydrophobic part. So anyway, this barrel, right, and this kind of gets to the properties of amino acids. The barrel along the outside, right, this is the part that's in contact with the inside of the membrane. That part has to be hydrophobic, because otherwise it will not be able to exist in that place, right? You can't have a hydrophilic thing in a hydrophobic environment. It just doesn't work, right? So the outside of the barrel has to be hydrophobic. The inside is what passes protons from the intermembrane space into the matrix. And so that has to be hydrophilic, right? Because otherwise those protons won't want to move through, right? So just in the space of, one part of a protein, right? And this is essentially, I think there are multiple proteins, I can't remember how many, to be honest, that make up this part. It has to be both hydrophilic and hydrophobic in order to be able to assemble properly and do its job. And then the cool thing is this actually physically rotates, and that rotation is what generates ATP. That's what makes it super cool. But anyway, I just thought I'd show you this because it kind of illustrates all the different properties that a protein must have for it to work. And those properties come from amino acids. Okay, so now we're going to get into the translation part, the actual mechanism of translation. And there are a bunch of actors, essentially, right? We've already talked a great deal about mRNA and whether it's prokaryotic or eukaryotic. And now we need to talk about the other things involved, right? So the main one, at least in many people's mind, is the ribosome. Oh, I'm sorry. Friday morning blues. And the ribosome consists of, I'm not sure, hundreds of proteins, but almost an absolute ton of proteins and ribosomal RNAs, right? And these are grouped into two subunits, right? We've got the small subunit. This is what binds to mRNA, and it also has ribosomal RNA in it too, which I didn't know, which is kind of cool. And the large subunit, and this is where the peptide bond formation occurs. And so these sites are where tRNAs come in and drop off their amino acids and then leave, right? And so essentially we have three different sites because there are three different things that need to go on. One is the one where tRNAs come in, right? And they're going to recognize the mRNA, their codon, that is currently in the A site. So this actually stands for amino acyl tRNA binding site, which is a bit of a mouthful. So I just shorten it to the access site, right? This is where the tRNAs gain access to the mRNA. There's a P site, and this is the peptide bond forming site, the peptide tRNA binding site. And so this is where the peptide bond is formed between the amino acid on the new tRNA that's in the A site and the existing amino acid or amino acid chain that's connected to the tRNA in the P site. And then finally, we have the exit site, and this is where the tRNA that has been discharged, right, doesn't have any amino acid attached to it anymore. That's where it leaves. And so what the tricky concept to get is that there's this constant flow through of tRNAs through this ribosome, through this large subunit, but that the whole ribosome moves along the mRNA, right? So the tRNA that comes into the A site gets the polypeptide chain transferred to it. The ribosome moves one. That tRNA is now in the P site. Another tRNA comes in. Peptides have moved over. Ribosome moves one. That tRNA is now in the exit site, and now it leaves. Right, so there's this constant flow through of tRNAs coming in, dropping off their amino acids, and then leaving to be recharged by the next protein that we're going to talk about. And the catalytic activity that is required that does that, essentially the formation of the peptide bond, which we talked about further up, that's the one. Sorry, a little bit bunged up today. That's the one between adjacent amino acids, the C on the first one or the prior one, and then the nitrogen on the second one. And so that activity and also the cleavage of the amino acid from the tRNA, that's carried out by ribosomal RNAs, not the proteins, right? So the main work done by the ribosome is done by RNA. It's not actually done by the proteins. Proteins provide the shape and the space and the like for that to happen. Now, the next important player is the tRNA, right? And there will be 20 tRNAs, essentially one for every single one of these. Actually, more than that, one for every single codon that encodes a amino acid, right? And these are RNA structures, right? They don't encode information, well, not really, and they don't have any catalytic activity. Their job is to bridge the gap between the mRNA and the information in it and the amino acid sequence of the protein, right? And they do so by having, and they're always drawn like this kind of cloverleaf shape, but they really look more like this. Which is a kind of, I always kind of think of them looking a little bit like elf hats, to be honest, but that's just me. Anyway, they have a lot of secondary structure. So remember we were talking about how RNAs can bind to themselves to form double-stranded RNA. Well, this is double-stranded RNA. These are a series of stem and loops, right? And I'm not really sure what these ones do. They probably have a role, particularly as they have conserved bases. They have a role in something in translation. I just don't know what. And this end is where the amino acid is attached. So this is essentially it'll be fairly reasonably large, kind of about that sort of size compared to the tRNA. And this down here is the really important part. This is the anticodon, right? So we talked about anticodons, right? I'm pretty sure we did. Tell me if I didn't, because it's really important. Well, okay, I'm going to assume that I didn't then. Oh, we did. Great. Awesome. So this anticodon has the complete opposite of the codon that encodes the amino acid that it carries. And so this is the means by which that tRNA recognizes that codon, because the 3 bases here will be the perfect complement to the 3 bases in the mRNA. So if it's, for example, AA in mRNA, you'll have UUU in the tRNA, and so on, for example. It'd be the complete opposite. And this is how tRNAs translate, essentially, or facilitate the translation of that information that is recognized by the anticodon into an amino acid sequence. Now, you're probably wondering, okay, well, what determines the specificity of that amino acid, right? Because perhaps you could have any amino acid here, right? In which case it wouldn't be very specific. So that specificity is maintained or created even by the thing that adds the amino acid to the tRNA. You don't need to shut the door, love. And that's a protein called amino acid tRNA synthetase. And there is one of these for every single amino acid. And so these have specificity on 2 levels. One, what amino acid they will bind, right? And that's very specific to the shape of the amino acid, right? And you know that they're very different in their shape and their properties, charge, hydrophobicity or philicity, right? They're very, very different, each and every one of them. And so the enzyme recognizes the amino acid, only the correct one will bind. And then it's basically kind of kept there, right? And it's kept there with the help of the energy of ATP, right? So ATP is hydrolyzed and actually becomes AMP, which is kind of crazy, right? Usually it just goes down to ADP, but in this case it's AMP, which is adenosine monophosphate, right? So there's just one phosphate involved. And this kind of promotes the retention of the amino acid. The tRNA comes in, and now we get the second aspect of specificity. Hey, Lily, come over here. One of our puppies got adopted, and so the other one is all out of sorts. Here she is. Come on. Can you see her? Isn't she batty? Come on, up. Up. Oh, I've got to get her up here, otherwise she's just going to go nuts. Come here. Very skittish. You can get close up. She's called Lily. She chewed up a set of my wife's headphones the other day, which I was kind of mad about. Aww. All right, little puppy break is over. Now off she gets. Crazy. She's missing her little buddy that she was fostered with, who went on her trip to New Jersey, amazingly. A lady in New Jersey adopted her, so she went yesterday, she's probably still on the road actually. It's a 2-day trip, which is kind of crazy. Yeah, I know, but that lady actually had already adopted, I think, 2 dogs from this rescue center that we help, which is kind of nuts. But anyway, she seems to really love her dogs, and so she'll be in a good home. Shame because she was super cute, like this little black ball, with like really, really short legs, about that long. Anyway, so yeah, hopefully Lily will go find a patch to lie down on and be quiet so I can concentrate. Because she's very distracted. She's super, super cute. Anyway, and she'll be gone soon, actually. She's just been adopted by another family. Not sure where. Here, I think. But again, don't know. So she'll be out the house, soon. We'll be back down to 1 dog and 3 cats. Be very quiet. Anyway, so once that amino acid is bound and kind of kept in place with the AMP, now tRNA comes in. And this is where the second specificity bit comes in. Only the tRNA with the correct anticodon will be able to enter this region and bind to the transferase or the synthetase. So not only do you have the correct amino acid here, but also you screen out all the wrong tRNAs by only letting the one with the correct anticodon bind to that protein. And this is kind of a computer model of what we think that actually looks like based on structural studies, essentially. Right, and so you can see how the amino acid is kind of held in a pocket in one of the folds, and then we have the anticodon down here in its own little pocket. Now, when the tRNA comes in, that displaces the AMP, and the bond between the amino acid and the tRNA is formed. As soon as that happens, Then the charged tRNA, right? So this is the tRNA with amino acid on it, or amino acyl, then leaves, and this process can happen again. So there's this continuous cycle between tRNAs getting charged with amino acids, going to ribosomes, dropping them off, becoming discharged, right? Coming back out of the ribosome, getting a new amino acid added to them in a constant cycle, right? And so you can imagine that this takes a lot of energy, right? Protein synthesis is very energy intensive, not just for the formation of tRNAs with the correct amino acids. You have to make the amino acids or get them from your diet, right? We get the majority we make, but a large minority we have to get from our diet too. And all of the energy involved in the actual translation process as well. So it's a very energy intensive process. And actually, this is one of the, if you kind of enter starvation conditions, this is one of the processes that starts to shut down, right, to conserve energy for, organismal survival. Okay, so essentially, as with transcription, we have initiation, elongation, and termination in translation, right? So we have to start the process continues, and it continues until it's told to stop, right? And again, just as with DNA polymerase and kind of with RNA polymerase too, there's nothing intrinsic in those enzymes that knows when to stop, right? They have to be told to stop by either sequence features or other things, right? That tell them to finish the job. And so when we talk about initiation, now we get two big differences between prokaryotes and eukaryotes. So initiation of translation in prokaryotes is pretty straightforward. Essentially, we have our two subunits and they want to associate, right? They want to kind of get together, and we need to prevent that from happening until the small subunit binds to the start codon, right? So this is facilitated by an initiation factor called IF3. This binds to the small subunit, stops the large subunit from assembling, and also helps recognition of this sequence here, which is called the Shine-Dalgarno sequence. And so this is a short sequence in the mRNA which tells the small subunit this is where the start codon is, right? And so it assembles right over the top of the start codon, and in such a way as well that the large subunit, when it assembles, the P site will be over the start codon, right? So the middle site here, so this reads in order of action, it reads ape. And I don't know why it's always put that way around. I don't actually know. Perhaps because we always think of, RNA going from left to right, 5 to 3 prime. Could go the other way too, right? 5 to 3 prime that way. Doesn't actually matter. I don't know why this is always put this way around. But anyway, so the small subunit assembles directly on the start codon. And then we have a whole bunch of stuff join, right? So we have GTP, right? This is a alternate form of energy, kind of analogous to ATP. Quite often used in metabolism as well, these kinds of things. We have a tRNA charged with methionine, right? So this has our anticodon that will recognize the start codon. And we have a few other bits and pieces that will come in and help that process. Right, so we have this whole assembly that basically brings in and complexes around the start codon. Then, once that's in place, now the IF3, right, the thing that prevents the large subunit from binding, and all the other jazz, and that's also facilitated by the catalysis of GTP to GDP, right? So this is, again, the same as ATP being broken down to ADP, basically releases energy. That dissociation of all of this stuff allows the large subunit to assemble, right? And as soon as the large subunit is on the small subunit, right, now we have a functional ribosome, and now we have our next codon is in our A site, right? So essentially we have our methionine tRNA in our P site, and now our access site is open. Now another tRNA could come in that recognizes that particular site, whatever UGC happens to encode. I don't know off the top of my head. Anybody got any questions? No? Okay. And as soon as the first, well, not the first, the second codon, the tRNA comes in, that methionine gets transferred to the, essentially, the start of the other amino acid, or kind of a little bit complicated to describe. Then the whole ribosome moves along one codon. Now this discharged tRNA that had methionine but doesn't anymore is in the exit site. It leaves. The second tRNA with 2 amino acids, methionine and then whatever this one encodes. Actually, do we have a codon table in here? We do. I keep saying whatever it encodes. UGA is, hang on, that can't be right. UGC. Phew, I was going to say UGA is a stop codon. UGC is cysteine. There you go. It kind of annoys me to keep saying whatever that is, right? It's easy enough to just find out. All right, so then you'll have a tRNA in the P site, which has methionine and cysteine, 2 peptides, so a dipeptide. The access site will now be free. Another tRNA comes in for whatever the third codon is. Those 2 amino acids get transferred over. The whole thing moves along again. And then the second tRNA is now in the exit site and it leaves. And so this is constant kind of coming in, transfer amino acids, ribosome moves along, tRNA leaves. tRNA comes in, transfer amino acids, ribosome moves along, tRNA leaves. And it just keeps doing that, all the way along. And there's, let's see. Here we go. Let's just, I'm not going to get you to watch this because sound doesn't quite work super well. But this is a, no, thank you. Neat video. It doesn't have any commentary, right? So it's not a great one for learning the individual bits and pieces, right? Which is what we're talking about here. But it does very nicely visualize that kind of constant flow through of tRNAs through the ribosome as the ribosome processes along the mRNA. So you're probably thinking, oh, that's all kind of complicated. Well, it gets even worse with eukaryotes. So we're going to go through initiation for both eukaryotes and prokaryotes, or prokaryotes and eukaryotes first. And then I'm not really going to spend much time on elongation because, it's fairly straightforward once you kind of get it straight in your head. And then we'll go on to termination of translation. So in eukaryotes, things are quite considerably more complicated. So in prokaryotes, we have a plonk straight down on the start codon kind of deal. In eukaryotes, a lot of other parts of the mRNA are actually involved in translation, right? Not just the start codon. And also some of the features that we've talked about are also heavily involved. And so essentially the main takeaway is that the ribosome actually starts in the 5' UTR, right? It doesn't assemble over the start codon. Part of it assembles on the cap and scans along basically until it finds the start codon, and then assembly and translation proceeds, which is kind of what we have here. Anyway, so some of the regions that we talked about before, which are super important, are the 5' cap, right? The methylguanosine cap that is added to the 5' end to protect it. This is very, very important for the initiation of translation, because this is recognized by the cap binding proteins or elongation initiation factors, which we'll see in a little bit. The 3' UTR and poly A tail is very important too, pardon me, because the 3' UTR bends round, right? So it physically gets bent back so that the poly A binding proteins, right, which we talked about before in terms of stability, help recruit and retain that ribosome at the 5' end of the 5' UTR. Right, so all of this is important in initiation of translation, right? And obviously there's lots of ways in which we can interfere with this if we want to control how effective translation is or how frequently it begins. Okay, so that's the general overview, right? We have actually it's not the whole ribosome. This is a little bit inaccurate. It's a bit too much of a simplification. We have our small subunit recruited by cap binding proteins and aided by the poly A binding proteins on the poly A tail. Okay, so in detail, however, it gets way more complicated. And so the general principle here is that of a scanning process. And so the small subunit scans the 5' UTR using a methionine tRNA, essentially is like a barcode reader, right? And so it has that, and it kind of moves along until that UAC, that anticodon, recognizes it's matching AUG, or the start codon. And as you can imagine, there are no other AUGs in this region, because otherwise it would not work right. You know, we get the initiation and translation in the wrong place. Okay, so the very first part is a bunch of proteins. These are the cap binding proteins from the previous slide, assemble around the 5' cap, right? So they recognize that very particular shape and structure of those methylguanosines that are attached to the 5' cap. And this 5' UTR can be anywhere from 50 base pairs to several hundred base pairs long, right? Varies from gene to gene. So once these have assembled, now we can start assembling all the other business involved in that barcode scanner. So we have obviously our small subunit. This is just its size in basically how quickly it falls through a sedimentation gradient. So that's what these S numbers mean, and that's why they're different. They're basically a rough measure of size. So we have a RNA and a ton of other proteins, right? Just making up this one piece of the puzzle. And this is helped by a bunch of initiation factors, right, which recognize the cap binding proteins and also bring in this tRNA, which has a methionine on it, right? So this is our initiated tRNA. And I think it's Pretty sure you don't ever find methionines elsewhere in a protein. I can't be 100% sure about that, but they're very, very rare, to the point where I think you only find them at the beginning. But I might not be 100% correct about that. Obviously, because I don't know every protein sequence, but from memory, that seems to ring about true, right? So this is a very specific tRNA for initiation, right? You don't tend to find it elsewhere in the protein or that codon elsewhere. Okay, so all of this whole jazz assembles includes the initiation factors. We have our small subunit. We have our tRNA with methionine, and those are all bound to and recruited by these cap binding proteins. And then I believe with some help from the poly A binding proteins, this whole complex is released from the 5' end and begins to process or scan along the mRNA until it finds the stop codon. Okay, so it kind of chunters along, probably listening to some nice music, checking out the sites, wait until it finds an AU gene. And as soon as it does, all of these other initiation factor proteins are released with the help of another one. It kind of comes in. And the large subunit over here assembles, again, the same actually as in prokaryotes, with the tRNA for the methionine in the P site. So now we have our whole ribosome, which consists of the large and the small subunit, and a tRNA in the P site. So now we essentially have the same deal occur, right? And thankfully they tell me what that amino acid, so I don't have to look it up. Our next codon is UUU, right? That's recognized by a tRNA with an AAA anticodon. And that carries phenylalanine. So that comes into the A site. This methionine is cut off and attached to the phenylalanine. Then the whole process moves along. And now this tRNA, which was in the A site, is now in the P site, has two amino acids attached to it, methionine and then a phenylalanine. The discharged tRNA for methionine is in the E site. It now leaves, essentially goes back and gets recharged by the amino acyl synthetase for methionine. The A site is open, and now it's going to be over the next codon, which here is GAA. So the next one would be, come on, GAA, which is glutamine. I've forgotten how to read those things. How embarrassing is that? So now this whole thing is moved along one set of 3 letters, one codon. Now we have our tRNA for phenylalanine in the P site. Our tRNA with glutamine comes in. No, glutamic acid, sorry. Not glutamine. GLU is glutamic acid. Comes in. Those 2 amino acids get added to the glutamic acid in the A site. Whole thing moves along. This tRNA, which had phenylalanine, is now in the E site. It leaves, gets recharged somewhere else. Now the A site is over a new codon, right? So it basically moves along in these jerks of 3 bases at a time. Every time it moves along, the existing amino acids are added to the new one. A new tRNA comes in with amino acid. An old tRNA without an amino acid leaves. Peptide bond formed, moves along one. New one comes in, old one leaves. Peptide bond forms, moves along. Over and over and over again. That's essentially the process of elongation. And these are also catalyzed by different elongation factors, right? So not the initiation factors that we saw up here, right? The IFs. Now we have a whole bunch of EFs, or elongation factors, which basically bring in GTP to power this process, and then release that GDP from those, and then kind of constantly do that jazz. And I don't know if this, there we go. So this gives you a real nice kind of view of essentially the mechanism by which this occurs. And this is by, from what I can see, this is in prokaryotes, because we have the shined organo sequence here. So this is well worth a look through. Kind of, it's a good way of explaining the nuts and bolts, essentially, of this process. Okay, I'm going to leave you to do that yourselves. Okay, and so just to finish up, let's post that in the chat, just in case people want to watch that from there instead. Okay, and just to finish up, because this is kind of a nice place to stop, literally, right? So this is termination. So, oh yeah, here's what I thought it was. So essentially what we have in our codon table, right? So this is all of the different combinations of 3 letters that you can get from RNA. And within that, there are 3. So all of these correspond to an amino acid, right? So these are our codons here, and next to them are our amino acids like threonine, proline, glycine, and so on, right? And here's our initiator start codon, initiator tRNA for methionine. Now there are 3 which do not encode any amino acid, right? So there's no tRNA which will be able to recognize these and continue the process of polypeptide elongation, right? And so essentially the ribosome stops because there's nothing there, right? So there's no tRNA that can come in and continue the process. And so all the way along, right? So we're basically kind of adding our amino acids to this chain, right? So essentially what you see here is the second to last tRNA. It's having its polypeptide chain kind of cleaved from it using GTP hydrolysis to power that. And the ribosomal RNA is formed the peptide bond between that one, which is a lysine, and asparagine, right? These last 2. And so now everything, that whole polypeptide chain is attached to this tRNA. The whole thing moves along, and that's now in the P site. Now, if there's nothing in the that can come into the A site, right, because there's no tRNA. I don't know who that is. Stuff that underneath the couch. Now what comes in is something called a release factor, right? And so this comes in instead of a tRNA. And it kind of, it's not entirely clear how this works, but essentially GTP helps release the polypeptide from that last tRNA, and also kind of kicks this release factor out and dissociates the whole ribosome. Right, so again, the process is the ribosome pauses with a stop codon in the A site, a release factor comes in, and then GTP hydrolysis both releases the finished polypeptide and also dissociates this complex of the ribosome and the release factor as well. these are then able to resume the whole process, right? This small subunit will be able to recognize another 5' cap binding proteins, right? It'll be able to assemble there, and the tRNAs will be able to be recharged. Polypeptide goes off and gets folded, and also the large subunit kind of hangs around waiting for another small subunit to kind of call out to it and say, here, I'm ready to go. So that's the whole process of translation. And what we'll do on Monday is just talk a little bit about the genetic code and some funkiness in prokaryotes, basically. And that should kind of maybe not even take the whole class on Monday. So if over the weekend, if you can, go through any of the material that we've covered so far and start making a list of stuff that you want to ask. And we'll be able to use a little bit of time on Monday, for pretty sure, and the whole of Wednesday to go through those questions. So you can use those as a study session for the exam on Friday. Okay, and once I've got off here, I'll change that schedule and post it up and let you know as well.
I have a question, Dr. Crook.
Hello.
The PowerPoint that you have listed for this is chapter 16's PowerPoint, correct?
Ooh.
Right, that's what it says at the very top.
Oh yeah, number 16. I don't know, it doesn't correspond necessarily to chapters in the book though.
Okay, well, the PowerPoint that you have posted in Blackboard, it varies. It's a little bit different.
Oh, okay. I'll upload a new version.
Yeah. Will you just make sure that it is right up to date?
Mm-hmm. Yeah, I think the main difference is I've swapped a few different things around between this one and the prior one. So some of the order was a bit off for some reason.
Okay, well, I think, and I don't think it's just the order. I think some of the content isn't in there. For instance,
okay.
the amino acid chart was not in there.
Oh, that's weird.
So maybe just redo that, just because I know I'm probably not the only one. It's probably everybody. So
no, that's great to know. And kind of a little bit weird as to how that kind of occurred, because I try not to change things, right? Because that causes these kinds of problems. But yeah, I'll go through and double check and upload the latest versions that we've been using in class. Okay, so you know that those are the ones to study from. Cool, thanks for the heads up. All right. And as always, if you have any questions about any of this stuff, just shoot me an email as well. I'm very happy to reply to those. Cool. All right. Thanks very much, everybody. Have a lovely weekend. And I will see you Monday. Oh.
Bye.
Let me guess. Is he or she called Snowball?
No, Lotus.
What kind of dog is that?
It's a half Maltese, half Shih Tzu.
Oh my gosh, my mom has a Maltipoo, so half Maltese, half poodle. So she looks kind of like that, but looks a little bit more like a poodle. But they're about to get an all-white standard poodle for my best friend, whose brother bred them. So.
Yeah, she is a puppy of my dog. So
nice. That's awesome.
Super cool.
My Siamese cat is pregnant, so we're expecting babies.
Oh, that's so exciting.
Yeah, I'll have to show y'all when the babies come.
Yeah, for sure. I'll make sure at least one of my kids is around to show them, because they're nuts about cats.
Yeah.
Oh, what a calm dog.
She is. She's the laziest one. That's why we kept her.
Oh, very cute. Thank you for the showing.
Okay, we all have a good day. Bye.
You have a good day.
You too.